Hello students, the topic of today's video lesson is regulation of gene expression. Genes are present on the DNA and these genes are transcribed, then translated into proteins. So how it is possible? Suppose we have a gene sequence T, A, C, C, T, T. So this is the gene sequence present on the template strand of double-stranded DNA. Clear? So first step is the transcription. So it will transcribe and it will produce the messenger RNA. So the gene sequence in this messenger RNA will be adenine, then uracil, guanine, then G, then adenine, then A. Okay, so this will be the sequence in the messenger RNA and these are the genetic codes. Now the next step is the translation. So this is the second step, translation, where these genetic codes will converted into the amino acid sequence. So AUG, it will code for methionine and this codes for glutamine. So like this we have the chain of amino acids. This is known as polypeptide chain. So finally the conversion of this gene sequence into the polypeptide chain. Conversion of gene sequence into the polypeptide chain. This is known as gene expression. Okay, so this is the gene expression. In each and every cell that is directly affected by the environmental, physiological and metabolic condition, and they respond according to these conditions. Suppose this is a cell, it is growing in a nutrient medium. So any change in its environmental, physiological or the metabolic condition, it will lead into the production of different types of enzymes or proteins. Okay, so there is the change in the condition, change in the condition. This change in the condition will leads to change in gene expression. And finally, we have different types of products. These products, they will help the cell to cope up with the changed environmental condition. Okay, so these responses are the result of the products produced by the gene expression. Hence we can say the gene expression is also affected by the conditions, different type of conditions. In eukaryotes, the gene expression is regulated at several different levels. Number one is the transcriptional level. Second one is the processing level, transcription level, where during the conversion of DNA into messenger RNA. Then the processing level, where the heteronuclear RNA, it is processed and converted into mature messenger RNA. Second is the transport of messenger RNA. As we know, the messenger RNA is synthesized in the nucleus and it is transported into the cytoplasm for translation. So third control is at the transport level. Then fourth one is the translational level, where the messenger RNA is converted into the polypeptide chain. So these are the several levels in eukaryotes where the gene expression is regulated. But in case of prokaryotes, the initiation of transcription, that is the first level, it is the main site for the control of gene expression. And in our textbook, we have to cover only this topic, that is the regulation of gene expression in prokaryotes. And we have the example which we have to study in detail in our class 12th is the Lac Operon model. This model was proposed by Jacob and Monod in 1961. Regulation at the transcription level. In the Lac Operon model, the word lac, it refers to the lactose. What is lactose? It is the carbon source or the energy source. And the word operon, operon refers to a polycistronic structural gene. What is the polycistronic structural genes? Means there will be a single messenger RNA, and this messenger RNA will produce different type of products. Product number 1, product number 2, product number 3, etc. after translation. So this is the polycistronic structural genes, and these are regulated by a common promoter. This system is known as operon, and operon is very common in bacteria. Clear? This is the operon, structure of the operon. Operon have 3 elements. Number 1 is a set of structural genes. Here the Z, Y and A. These represent the structural gene. The Z, it encodes for beta-galactose. Then the Y gene encodes for the permease, and A gene encodes for transacetylase. So these are the structural genes. Clear? This is the operator site for these structural genes. An operator site which is an DNA sequence that regulate the transcription of structural gene. So it regulates the transcription of structural genes. P is the promoter site, and I is a regulator gene. Okay, here the word I means the inhibitor, which encode a protein that recognize the operator site. It will produce a messenger RNA. Messenger RNA will produce a protein, and this protein will recognize the operator site or the operator sequence. So this is the structure of Lac Operon in bacteria. Okay, now how the bacteria generally prefer glucose as the carbon source to obtain the energy. But when lactose is also present in the environment, in that condition the bacteria synthesize the beta-galactosidase along with permease and transacetylase. Why these are needed? Because these enzymes will break down the lactose into glucose and galactose. Okay. So when lactose is present in the medium, then there is a need to transcribe these genes. Okay. And if lactose is not present, then there is no need of these enzymes. So the gene will not express, but in this condition the gene expression will take place. Okay, so lactose act as the inducer and it regulates the switching on and off of the operon. So lactose, if it is present in the medium, it will on the switch. If it is absent in the medium, then it will off the switch of operon. And when the switch of operon is off, then the gene Z, Y, and A, they will not expressed. Okay, so let us see it in detail. So the condition number 1, when lactose is not present in the medium. In that condition, the repressor is synthesized all the time from the gene I. This is the condition that repressor will be produced by this gene I. So it will produce or it will synthesize in the cell all the time. Okay, so this is the repressor messenger RNA, and this repressor messenger RNA will produce the repressor protein. Okay, now this repressor protein, it will bind to the operator region. Here it will bind here. So binding at operator site will stop the transcription, means the transcription process will not initiate. Okay, so the transcription of this gene will not take place and the lactose cannot be metabolized. Okay, so gene A, Z, Y and A, they are not expressed. This is the condition when lactose is not present, and actually there is no need of these products. Okay, hence it will stop. Then the second condition, when lactose is present in the medium. When lactose or allolactose, it is an alternative form of the lactose, when they are present in the medium, in that condition, so this is the repressor, it will produce the repressor messenger RNA, it will translate it and produce the repressor protein. Now the condition is when lactose is present, then lactose will bind with this repressor protein like this. Okay, so it inactivates the repressor. This is inactivation of the repressor. Now the operator side is free, so the messenger RNA produced here will transcribe these structural genes. Okay, so we have the messenger RNA, and this is a polycistronic messenger RNA, so it will produce 3 different types of products. Here it produces β-galactosidase, the product number 1 from gene Z, product number 2 from gene Y, this is the permease, and product number 3 transacetylase from the gene number 3. So we have different types of enzymes produced from this single messenger RNA. Clear? So now these enzymes, they will metabolize the lactose and in a normal manner. Clear? So here the regulation of operon is controlled by this repressor. So this is known as the negative control or the negative regulation. Clear? So this is all about the Lac Operon.